Good evening, everybody. I think it's probably a good time for us to get started. My name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad Institute, and it is my pleasure to welcome you to tonight's Science for All Seasons talk, the theme of which is going to be the next phase of big genetics. We are joined by Alicia Martin, Amit Khera, and JT Neal. Alicia is a population geneticist and a postdoctoral fellow with our program for medical and population genetics, and also at Mass General Hospital. Amit is a cardiologist at MGH and is also an associated scientist with our cardiovascular disease initiative. And JT is a cancer biologist with our cancer program. Tonight, they're going to talk to us about genome-wide association studies, these massive studies that study the genetics of thousands of people at a time. What these studies have taught us about the biology or the genetics behind human traits and human disease, how what we're learning from these studies is helping us to figure out how to better predict people's risk of disease, and also the challenges of translating all this genetic data into biological insights. I would ask that you hold any questions that you might have for the Q&A session that we'll be holding after JT, Alicia, and Amit have all spoken. And with that, Alicia, you're up.
Hi, so I'm happy to kick this off, and I'm happy to talk about diversifying genomics, so the who, the why, and the how for genetic studies. So I want to start really, really basic to make sure we're all on the same page for understanding the human genome. So as you're all likely aware, and you've heard probably countless times, maybe in your high school biology classes, human genomes between pairs of individuals are about 99.9% identical. And of course, that sounds like a lot. So all of your A's, your T's, your C's, and your G's on average are matching the person sitting to the left or right of you. But the human genome is really big. There's about 3 gigabases in the human genome, which means on average between any pair of individuals, about 4 to 5.5 million sites differ between pairs. And all of those variable sites are in every single one of your trillions of cells in your body. And it's these 4 to 5.5 million variable sites that make up and shape this heritable physical and biomedical variation. So if we look across the globe, we can see some plainly visible variation that's really heritable, that's passed on from your family members. So for example, looking across the globe, you can see differences in genetic variation that contribute to things like height and skin color. There are a bunch of different types of traits that we might be interested in understanding the heritable basis of. So there are, on the left side, some single gene or relatively simple traits. Things like, do you have freckles? What color are your eyes? And what is the Mendelian basis of cystic fibrosis? Do you have this disease? Do you not? Same with things like Huntington's disease. But most of the diseases that we might be interested in, and a lot of the complex variation, comes from the entire genome. It's the aggregation of many complex sites across the entire genome that are contributing to the heritable basis of these traits. So things like height, weight, skin color, and all of these various traits are made up of many, many loci across the genome. So as you know, your DNA is inherited from your close family members in time. So looking at this plot, we can see that with our close relatives, so looking at your mom or your dad, for example, you share about half of your DNA with them, and it's passed down from generation to generation. If we look further back in time, you can see that there's smaller proportions of your DNA being shared. So you get about 1/4 of your DNA from your grandparents, about 1/8 of your DNA from your great-grandparents, and so on and so forth back through history. And so this looking back through history is really important for thinking about how we're related to different populations. So looking through our family trees back into history tells us about how much DNA we share with our ancestors. So someone of the same population as you likely shares more genetic variation than someone of a different population than you. So in thinking about human history, our genetic differences are informed by this whole history. So humans originated in Africa, and then they migrated out of Africa about 50 to 100,000 years ago to populate the rest of the globe. So they migrated into the Middle East, and then into South Asia and Melanesia, into Europe, into East Asia, and into the Americas. And every time they migrated, they brought a subset of diversity with them. So the most globally diverse populations on the planet exist in Africa, and then humans brought a subset of that genetic diversity with them as they migrated out of Africa and into the rest of the globe. So this pattern is called a founder effect, where every time humans migrate, they bring a subset of that genetic diversity with them. And these differences across humanity also make up the correlation structure of the genome. So individuals who are more closely related share a more similar correlation structure in their genomes. When we want to conduct a genome-wide association study, so now comparing this genetic variation and things we can learn about population history with a phenotype, we run what's called a genome-wide association study, or a GWAS. So here we're testing whether a mutation is associated with a phenotype. And we do this time and time again. So I mentioned that we have millions of variable sites. We want to ask if every single one of those mutations is associated with a phenotype. So basically, it's just a correlation between a genotype and a phenotype. And we might adjust for things like your age, your sex, and importantly, as I've been mentioning, your ancestry. So as you all know, genetic studies have been growing larger and larger over the course of the past decade or so. The Human Genome Project, as many of you know, was completed in about 2003. It cost $2 billion to sequence a single human genome. The cost of sequencing a genome now is less than $1,000, so it's incredible to think of this as a feat of humanity in accomplishing. As a consequence of that acceleration in technology, our studies have been growing ever larger, and we've been gaining more and more deep insights into genetic variation and its contribution and its role in disease. So taking these studies and looking over the course of this time, what we can see is that we now understand a lot of the fundamental genetic basis of many different types of diseases, and we understand how complex they are. We also know which cell types contribute to those different diseases, and we can even look at which genetic mutations are contributing to disease such that we can learn about therapeutics. So some drugs have actually emerged from this last decade, this genome-wide association era. So that's a really exciting thing to watch this field grow so explosively over the course of the last decade, now that we're studying millions of individuals, where before we were studying hundreds or maybe thousands of individuals. However, if we now fill in this graph with who's participating in these studies, coloring by population, and juxtapose this next to the world population, what you can see is that we have a really inequitable picture here. So right now, about 80% of participants in genome-wide association studies are of European descent, which contrasts really starkly with the global population, where European descent populations make up about 16% of the world's population. This is also a really big issue if we look at the proportion of individuals as a function of time. So looking at this bottom part of the chart here, you can see in about 2014 was where we sort of maximized diversity in our genome-wide association studies. And since that time, diversity is stalled or kind of slid in the wrong direction, such that we're in no way on a trajectory to potentially correct these imbalances. And this has really big potential health disparities impacts if we're trying to think about translating genetics into the clinic. So this is a fundamental question that most of my research revolves around, answering how do Eurocentric genetic study biases impact the generalizability of this knowledge? And I want to be clear, the causal genetic variants are by and large shared across populations. So it doesn't matter who your ancestors are. Fundamentally, the genetic variants that contribute to the disease are shared across populations. But as I mentioned, the correlation structure of the genome really differs based on who your ancestors are. And so predicting your risk of genetic disease from these correlated predictors mean that you get differences in accuracy depending on the population that's been studied. So coming back to this question, how do Eurocentric genetic studies impact the generalizability of our knowledge? One thing I'm interested in is genetic prediction. And so to understand genetic prediction, I've looked at this very large biobank called the UK Biobank. It's sort of aptly named. Most of the study participants are of European descent. And I asked, if we look across many different traits, how well do we predict these traits in non-European populations relative to a European population? And so this chart shows you on the y-axis the prediction accuracy relative to Europeans, and on the x-axis you can see these different populations. So relative to a European population, we're doing a much worse job in these other populations. So for example, we're predicting European risk of disease about 2.5 times as well as we are in East Asians, and we're predicting European risk of disease about 5 times as well on average as we are in European populations by only studying European populations. So just coming back to this concept, the causal fundamental biology is not really what's differing here. It's simply differences in mutation frequencies and in the correlation structure of the genome across these populations. And this potentially has really profound impacts. So genetic precision is really different across these populations, which is very different from what we think of in the clinic right now, where if you prescribe a drug, it doesn't really depend too much on which population your ancestors are from. In general, that drug should have the same mode of action, and it should roughly work the same. There might be some exceptions to this rule, but that's kind of the average expectation. So there are some really big systemic challenges with diversity and diverse studies in genomics. I've just named a few of them, but I want to be clear that a GWAS is not where our biology is ending, but this systemic issue with diversity in genomics goes all the way down to understanding the fundamental mechanism of disease. So our genotyping arrays, the tools that we use to ascertain different variants and their frequencies, are sort of biased towards European populations, as are our imputation panels. So these sequenced panels that allow us to fill in the gaps and understand what the missing variation looks like, those are really biased towards European populations. If we do a study in a non-European population, chances are we'll have a really hard time tracking down a cohort for replication to make sure our science is really reproducible. And we're disseminating results in genetic studies at a different rate depending on which population you're in. So that's sort of like the genetic study level, but thinking even deeper, if we're trying to understand the mechanism of disease, we really benefit from having tools downstream of that. So understanding gene expression is a really important tool that allows us to understand whether genotypes that are associated with gene expression might be impacting some downstream mechanism. Additionally, cell lines in cancer, for example, share a lot of the same biases as our genetic studies. So about 80% of cancer cell lines that we have here at the Broad are derived from patients of European descent. These biobanks are also really overwhelmingly European. So to dive into that a little bit more, this chart is showing you that genetic biobanks are overwhelmingly white. So I'm showing you this map where you can see the US here in green, Europe in light blue, Asia in red, and then these other continents that are actually not represented in this figure, which is actually not filled in, my apologies. But what you would see if these bubbles were filled in with the corresponding colors is that by and large, most of our biobanks are coming from either the US or from Europe. And on the x-axis, you can see the percent of non-white participants. By and large, the percent of non-white participants are coming from Asia. And in the US, we have two large cohorts and two large biobanks that have a decent representation of non-European populations. Those are the All of Us research program, which you may have heard about. This is a spin out from Obama's Precision Medicine Initiative. And then the BioMe Biobank in New York, given that New York City is a very diverse city, also has a very large and diverse biobank. But we still need to do a better job with diverse representation in these very large biobanks. So one project that I'm really interested in that's here at the Broad that you might also be interested in hearing about is the Neuro Gap Project. So this is a neuropsychiatric genetics in Africa project. And I think this is a really nice example of moving towards diversity in genomics. One reason I'm so interested in this is because one of my focuses is in psychiatric disorders, which actually do a worse job even than the average genetic study area. So in psychiatric disorders, on average, we've studied about 95% European populations, and the remainder of that is primarily coming from East Asia. So we're really doing a very poor job capturing the genetic variants that influence global diversity and that influence psychiatric disorders. So the Neuro Gap Project is really exciting in my mind because it's one of the largest genetic studies that will have ever been conducted in Africa. And so it's bridged a lot of really important gaps in ethics, in bringing large-scale genomics projects to the continent. And so I think that's really exciting. And to do this project, we're partnering with collaborators in Uganda, South Africa, Kenya, and Ethiopia. So these are really vastly underrepresented populations, and in some cases, they have quite severe psychiatric disorders. I'm also really excited about this project because I think it's paved the way for how you should do ethical research in diverse communities. So there's this research capacity building endeavor that people are taking very seriously in this project to do in-depth training programs so that African researchers get trained over the course of 2 years through online weekly classrooms, through workshops, and through doing research proposals on their own, so that when they get data back from this project, they'll be able to analyze their own genetic data. So I think this is really exciting and is going to be building up the next generation of researchers for genetic studies in Africa. Okay, so I've talked a little bit about inequalities in genetics and where this is heading. I think to move towards public health parity in genetics and start to translate a lot of our really exciting and interesting findings, we need to think about how to correct these imbalances. So one area that I'm working on and that some of my collaborators are working on with me is developing new statistical methods in genetics to account for population differences so that, for example, we can more accurately predict genetic risk of disease even if you're in a non-European population. So that's a really nice idea, but I think there are like some really big problems that are sort of larger than just thinking about new statistical methods, and so they're going to need larger answers to these questions. And so we're going to need a lot of help for incentivizing large and diverse studies beyond just European populations. So one area that I think we'll need some help in is in funding. So we'll need the NIH, for example, the Wellcome Trust, and a lot of the largest funders of biomedical research in the world to push some money and to divert this effort into diversifying genomics, which will both help scientifically, we'll learn new biology, and ethically, this will help us with translating these technologies in a more equitable way. With data sharing, I also think that we need to make sure that we're disseminating results fairly and early for every population, such that we have access to genetics results from diverse populations. And then we need incremental releases. So right now, there are diverse data sets that are already out there that have been studied, but it's actually really hard to get access to some of these data sets. So I think we've seen some disproportionate benefit from studying some diverse populations already, and these are useful even when they're a fraction the size of European population studies. I also see a way forward in capacity building for global regions that are new at genetic data analysis. They'll disproportionately contribute if they're already well-trained and well-equipped to do these genetic studies. So with all of those in mind, I want to say thank you. I'll take questions at the end, and I'll turn it over to Amit, who's going to talk in more depth about polygenic risk prediction.
Thanks so much, Alicia. And my name is Amit Khera. I'm a cardiologist at Mass General Hospital and a researcher here at the Broad Institute. And today, I want to use a heart attack as a disease model to really describe how we're thinking about genome interpretation. There are, of course, a large number of people to thank, but I'll highlight first Mark Chaffin, a talented computational biologist in our group, and Say Catharason, who heads the cardiovascular disease initiative here at the Broad, where we did this research. Secondly, institutional support from both Mass General and the Broad. And I think I've been remarkably privileged to be in this community where we can work side by side with clinicians, computational folks like Alicia, genetic counselors, and really getting all the relevant stakeholders under one building. I think it's one of the few examples where that exists actually in the world. So I thought I'd start with a clinical observation. And this observation was actually made by Dr. Paul Dudley White, who started our cardiology division over 100 years ago. And the observation was actually quite simple. It was published in a leading medical journal in 1951. And he took 100 cases who had a heart attack at a young age, patients coming to our hospital, and 146 control. Now, his experimental design was actually quite simple. He had a survey, and he said, did your parent also have a heart attack? And it turns out that a family history of heart attack was present in 27% of those who had had a heart attack and only 14% of controls. Now, this observation has stood the test of time. And even in my clinic today, I ask patients about their family history. And we know that if your parent had a heart attack, on average, your risk is about double compared to everyone else. But this observation actually also stimulates a much more interesting scientific question, which is, what are the discrete molecular determinants that are driving this risk that runs in some of these families? And it's only within the last 10 years or so that we really had the tools to get at that question. So if you think about it, how might your DNA predispose you to a heart attack, say by the time you're 50? And there are really two models. The first is what we call a monogenic model. And here, mono meaning one, it's a single mutation, meaning a rare mutation present in an individual that in and of itself might triple or quadruple your risk for disease. A second model is what we call the polygenic, poly meaning many. And here it's not one mutation, but actually many variants scattered across the genome. And as Alicia said, in between any two of us, there may be several million different sites where our DNA differs. Each individual variant may have a modest effect, but you might imagine that in aggregate, their impact could be more substantial. So the traditional approach to thinking about genetic prediction has really been focused on rare monogenic mutations. And as a cardiologist, I'm most familiar with familial hypercholesterolemia. This is a situation where there's a single mutation, meaning a single misspelling in folks' DNA code, and that mutation prevents them from clearing the dangerous cholesterol from their circulation. So from the time they're born, they have very high levels of circulating cholesterol. And on average, this leads to a tripling of the normal risk for heart attack. And it turns out that when we look at very large populations, it's about 0.4% of our population, or 1 in 250 individuals, who actually harbors one of these mutations. And so there's a large number of efforts to actually go out and find these individuals so that we can help them and prevent them from having a heart attack before it ever happens. But what about the remainder? And here, we were really interested in thinking about what if we repeated that Mass General experiment in 2019? And here what we did was we worked with collaborators at Yale to identify 2,000 people who came in with a heart attack at a young age, so age below 55 years, to any of over 100 hospitals across the United States. And we systematically compared their genomes to almost 4,000 controls who remained free of cardiovascular disease. And I said, okay, well, if a patient comes into Mass General with a heart attack at a young age, and I'm handed a whole genome sequence, what is actually the state of the art in 2019 for how I might interpret that genome with respect to their risk? And so first, as has been done previously, we started with these monogenic risk mutations. And what we found was that in about 2% of cases, we were able to find a familial hypercholesterolemia mutation. And this is very consistent with previous studies that we and others have done. On average, these mutations were associated with an odds ratio of 3.8. So they were about a quadruple a normal risk of having a heart attack. Now, you might remember that these individuals have a mutation that prevents them from clearing cholesterol from their circulation. So here I'm showing you on the y-axis is the observed LDL cholesterol, and then on the x-axis whether or not they had a mutation. And what you can see is that although there's some overlap, LDL cholesterol, which is something we routinely measure in clinical practice, is actually a reasonably good biomarker for the presence of these mutations. So if you had a mutation, on average, your LDL was 202, versus if you don't have a mutation, the average 124. So what about the remaining 98%? And we wanted to ask a question. Can we actually identify additional patients using not a monogenic model, meaning one mutation, but a polygenic risk model, including many different variants scattered across the genome? And so to get at this question, we really used as foundation a study that was published a couple years ago. And it wasn't 100 patients, but actually an effort to find 60,000 individuals from around the world who had had a heart attack and comparing their genomes to 120,000 controls. And so for each of these variants, we said, is the variant more common in the people who had had a heart attack? Those are actually risk variants. Or are they more common in those who did not have a heart attack? Those variants actually might confer protection. And so some variants might increase your risk by 1%, others decrease your risk by 2%, and so on. And really, the output of that study was just a list of 6.5 million rows, and each variant, what is the percent change in your risk for heart attack? So we said, okay, well, we have all this information. How can we really think about integrating all this information into a single number? And it turns out that a number of folks have developed new computational algorithms. And so we actually had we were agnostic to the approach. We tried a few different strategies. We did 4 different approaches to combining this information into a single polygenic score. And then we said, okay, among these 4 scores, which score actually performs the best? And here, as Alicia mentioned, we turned to the UK Biobank, 120,000 people where we had access to both their genetic information and also whether or not they had had a heart attack. And it turned out that in every case, the score that integrated information from all 6.5 million variants significantly outperformed scores that had a fewer number. And so it really was the case that the ability to take all the variants, weight them appropriately, and combine them into a single number was the most powerful in predicting genetic risk. We then said, okay, this is our best score. Let's see how it does in an external, an additional, totally independent population. So we actually tested the score in 300,000 additional people. And what you can see here is that the score is actually normally distributed in the population, just like your blood pressure or your cholesterol. So this is unlike monogenic mutations where you either have one or you don't. Here, some people have a low susceptibility to heart attack, others a high susceptibility based on this polygenic score, exactly as you might predict. Moreover, if we bin these individuals into percentiles, so here I'm showing you on the x-axis percentiles of the polygenic score. So each dot actually represents 3,000 people. And on the y-axis is the percent chance that they had had a heart attack by the time they were 60. It turns out that in the lowest percentile, the risk of having a heart attack was less than 1%. But in the highest percentile, it was actually 11% of individuals who had had a heart attack by the time they reached their 50s. So again, this single number, which we could quantify from the time of birth for only a cost of about $50, actually provides striking gradients in terms of risk as it manifests itself over the decades. So we took the score and we actually said, well, as I mentioned, traditional studies compare carriers versus non-carriers. What would happen if we actually arbitrarily said, well, the top 5%, those are actually carriers of a high polygenic score. And the bottom 95%, those are non-carriers. And it turns out that what we observed was that these top 5% carriers actually had greater than triple the normal risk, just as has typically been observed for those monogenic familial hypercholesterolemia mutations. So we used this cutoff and came back to these 2,000 patients. Remember, these are 2,000 patients coming to a US hospital with a heart attack before age 55. And as I mentioned, 2% had a monogenic mutation. And we said, okay, well, what percent of them actually had a high polygenic score? And it wasn't 5% like in the general population, but actually 17% of these individuals. So it's 10 times as common as those monogenic mutations. Yet despite that, the risk conferred was almost identical, 3.7-fold. So again, almost quadruple the normal risk for heart attack. In this case, conferred by a polygenic susceptibility. So I mentioned that for monogenic mutations, LDL cholesterol is a reasonable in discriminating carriers versus non-carriers. And it turns out this is actually not the case for high polygenic score individuals. Here I'm showing you on the y-axis again, LDL cholesterol. And what you can see is that although the carriers have an average 132 versus 124, the distributions are actually highly overlapping. So I couldn't differentiate these individuals if they came into my clinical practice. Moreover, it's not just LDL cholesterol, but actually any of our traditional risk factors. Here I'm showing you a comparison of the top 5% of the polygenic score versus the remainder of the population for things like family history, 44% versus 35%. Hypertension, 32% versus 28%. So as you can imagine, these traditional risk factors are slightly elevated, but none enough to be clinically useful, such that these individuals really are flying under the radar within our clinical practice. So as a preventive cardiologist, folks often come to me and they say, you know, almost everyone in my family seems to have had a heart attack, oftentimes at a very young age. And in some cases, this really leads to a sense of determinism or fatalism, where they feel like no matter what they do, they're doomed to suffer a similar fate. And it turns out, actually, at least for polygenic susceptibility for heart attack, DNA is actually not your destiny. And our group has shown over the last few years that both adherence to a healthy lifestyle and taking medications like statins, which lower your cholesterol, can actually markedly reduce your risk 40 to 50%, even and perhaps even especially in those with a high polygenic score. So because I'm a cardiologist, I focused the talk mostly on heart attack, but it turns out that this concept is actually broadly applicable to pretty much every common disease. So we recently applied this exact same approach using millions of variants to atrial fibrillation, irregular heartbeat, diabetes, inflammatory bowel, which affects our intestines, and breast cancer. And in every case, we were able to identify several percent of the population with more than triple the normal risk for these important diseases. So in summary, for the genome-wide polygenic score for heart attack, if you think about a monogenic model, it's again about 1.7% of people coming to our hospital with a heart attack. On average, the odds ratio is 3.8, so a quadruple the normal risk. Currently in our clinical practice, we need to do more to find them, but LDL cholesterol is what's currently being done to identify these folks. because we know that if we find them, we can help them with both lifestyle and medications to normalize their risk. By contrast, a high polygenic score is actually present in 17%, so 10 times as many people. Despite that, the magnitude of the risk conferred is identical. And as I mentioned, these individuals are currently undiagnosed within our clinical practice. But if we could identify them, both lifestyle and medication could really help us to attenuate their risk. So I think at its core, when we think about genomic medicine and the potential for genomic medicine to improve public health, we really want to think about how can we look at someone's DNA and identify high-risk individuals. Next, we want to not only identify them, but actually inform them in some way and disclose their high risk to them, and then ultimately intervene to decrease the risk of disease. So we're working with a number of partners, including Color Genomics and our genomics platform here, to really design a single test that would allow you to find whether you had a monogenic mutation and also your polygenic score for heart attack. And that's something we hope is available even later this year. And initially, we planned to do it as part of a research protocol because, exactly as Alicia mentioned, we're very interested in figuring out how we can make these scores equitable across racial and ethnic groups, how we can actually disclose these risks in a way that patients and even their healthcare providers would understand, because it's really a new phenomenon. And even more importantly, what are the studies that we can do over the next 5 years to really demonstrate that it's actually useful to find people and disclose the risk? How can we close the loop and really demonstrate an intervention that decreases the risk of disease? And that's something that's going to be a major focus of mine in the Mass General Center for Genomic Medicine over the next 5 years. So with that, I'll thank you for your attention, and I'll turn it over to JT.
Hi there, I'm JT Neal, and I am a research scientist in the cancer program where I run a research group. And I'm really excited to be able to talk to you tonight about something that I and others in the community think is one of the next big challenges in human genetics. And that is, how do we take this large amount of GWAS data that we've been collecting across GWAS studies, and how do we actually use this to gain meaningful insight into the biological underpinnings of human disease? So as you've heard, GWAS studies enable us to connect the genome to disease. And they do this by showing us variants that increase one's risk of developing a disease or having a particular trait. But in order to really connect the genome to disease, we need more than just a list of associated variants. If we want to really be able to think rationally about how to design drugs to treat specific pathways involved in disease, we need a few more pieces of information. So we need to know the cell types that are involved. We need to know, out of this large mass of associated variants, which variants are actually causal and which ones are just coming along for the ride. We need to know the genes that these variants are interacting with, which can often be quite difficult because most of these variants are actually not in the coding genome. And then lastly, we want to put this all together and ask how the variants in genes are altering the functions of cells and tissues in order to bring about disease. So this is a lot to cover, especially in a 15-minute presentation. So we're gonna take this at a high level, and we'll break this into 3 pieces. So first, I'm gonna tell you a few of the pieces of information that we'd like to have and a few of the tools we'd like to use in order to tackle this problem. Then we're gonna go through a totally fake ideal example where if we had all the information in the world and all the tools in the world, we could use them to connect the genome to disease. And then lastly, I'm going to tell you why that would never work in the real world and the challenges that we actually have to figure out. All right, so let's get started. So, of course, the first piece of information that you have when you start one of these studies is you have the genome. So you have the sequence of the DNA. And this sequence, of course, is represented in these variants that you found in a genome-wide association study. But you have more than just the sequence of the DNA. You also have the structure of the DNA. And so the average human cell has about 2 meters of DNA in it. And so the only way you fit 2 meters of DNA in a cell that's smaller than 100 microns is you have to condense it. You have to pack it really tightly. So the way the cell does this is it loops your DNA around these complexes called nucleosomes, which are made of proteins called histones. And then it packs it really tightly into these fibers, which then further condense into chromosomes. Now, this is really great for storage, but it's really bad for access because this DNA is packed super tightly. So there's really no way to get in close and read the genes that are inside. And so what we need, what the cell needs to do is it needs to locally unpack the DNA strand. And so the way that it does this is it uses chemical modifications to turn this structure that we call heterochromatin, which is silent and unread, into a structure that we call euchromatin, or which is active and can be read locally. And so the way that the cell does this is by applying chemical modifications to the histone proteins and to the DNA strand that just loosen things up a little bit locally. And so this enables you to get in there and to read the gene. And so these chemical marks provide an additional layer of information that's on top of the DNA code. And so we call this epigenetic information. So epi is the Greek prefix for on or over. And so this is a layer of information that sits on top of the genome. And we can use that information to answer some of the questions we want to answer. And so additionally, just one last note, and that is that DNA structure in 3 dimensions can also play a role. So if you stretch the DNA strand out linearly, you may see 2 elements that are quite far apart, and you would think that they're completely non-related. But in 3-dimensional space, the DNA twists and folds, and so it can bring 2 pieces of DNA that are really far apart into close proximity to interact, and I'll show you why that matters. So we've discovered a pretty wide array of these epigenetic marks, and through years of study, we've sort of figured out which combinations of these epigenetic marks denote which functional elements of DNA. Or put another way, these things serve as signposts that allow us to read the DNA the way the cell would read the DNA. And so you can see here I'm showing you sort of a pictorial representation of what these epigenetic marks might be with the stars. And so here you can see that by reading different combinations of marks, we can distinguish areas of the genome that are active, like this gene or this gene promoter, from this enhancer region over here. And to further distinguish these active regions from inactive or repressed regions. And so we can read this information locally, or we can integrate this information across the genome. And so now I'm showing you here on the bottom all 23 pairs of chromosomes, with the letters kind of a little off the side there, laid out end to end. And so what you can do is you can take all that you've learned at the local level and spread it out across the genome to make this map of activity. So this is all the places in the genome where it's open and active and where genes are being read. And so this is really cool because these activity maps are unique to individual cell types. And so the regions of your genome that are open and being read are different in the neurons in your brain, from the T cells in your immune system, from the beta cells in your pancreas, from the melanocytes in your skin. And so this is because, of course, these cells have to read different areas of the genome and different genes to perform their essential functions. This is also how you can have a single DNA template that's almost identical in all the cells of your body, but you can read it slightly different ways to make all the cell types that you have. So all of this information can kind of be integrated together to help you make predictions about variants in genes and functions. But what we'd really like to do, the last piece, is we'd like to be able to go back into the lab and test our hypotheses. So what we want are robust cellular models. So whether that be a 2-dimensional cell line that you can perturb the gene of interest, or you can introduce a variant of interest, and you can see a phenotype related to the disease you care about. Or if you want to get really fancy, you can make a transgenic mouse, where you alter a few base pairs in the mouse genome, and then you see if that mouse is more likely to develop the disease you're trying to study. Okay, so with all of these things in hand, let's enter a fantasy world together, where we now have all the information we could possibly want and all the tools we could possibly want. And let's deal here with a totally fake GWAS study. So here I'm showing you again the 23 chromosomes laid out end to end. And I've now superimposed the results of a type 2 diabetes genome-wide association study. So each one of these black peaks here represents a SNP that we've named as being positively correlated with the risk of type 2 diabetes. So how do we find the cell type from this? Well, I just showed you that individual cells have unique maps of activity. And so what we can do in this fantasy world is we can go to our magic book, in which we have all the maps of all the cell types in the body. And we can thumb through that book, and we look for a cell type in which the regions of the genome that are open and active tend to correlate with the genomic variants that we see. And so lo and behold, we flip through the magic book and we find pancreatic beta cells. And you can see here that these regions I've highlighted in purple represent the areas of the genome that are open and active. And they happen to superimpose almost perfectly with the variants that we found in our GWAS study. So what this tells us is that beta cells are probably the cell type of interest. So now let's zoom into one of these regions and see if we can figure out which SNPs are responsible. And sorry, I'm using the term SNP and variant interchangeably. So SNP is a single nucleotide polymorphism. So here we've zoomed in on this region, and you can see that there are 3 variants in this region that are labeled by the red lollipops. And so, as is often the case with a GWAS study, none of these are actually in a gene. So we don't know right away which ones of these may be causal. Maybe none of them are. But we can superimpose those epigenetic marks that I just told you about and begin to get an idea for what these other unhighlighted areas of the genome are doing. And so when we do that, what we see very clearly is that off to the left side, where we have our 2 variants clustered, this area is actually repressed or inactive in our cell type of interest. So it's very unlikely that these are the variants that are causal. Whereas if you go all the way over onto the right side, you can see that we have this SNP here sitting in an active enhancer region. So it's more likely than not that in this cell type, this SNP is the causal SNP at this locus. So now we'd like to tie that variant to a gene, because the SNP is not operating in isolation. It's interfering with some gene or pathway function. Well, what do? This variant is sitting way over here by itself, and it doesn't appear to be anywhere near this gene over here. Maybe it's interacting with another gene that's off the side of the screen I haven't shown you. Well, we can go back to our magic book, and we can look at the structure information that we have. And what we see is for this region, the DNA happens to be folded back on itself, bringing this enhancer region, which was quite far away, now proximal to this gene. And so we can pretty confidently now say that this variant that we found is interacting with this blue gene. So let's put it all together. So we have our cell type, our variant, and our gene. So to further extend the fantasy universe we're living in, we now have a magic freezer that we go into, which has any cellular model we could possibly want. And in it is a perfect model of beta cell biology. And so we pull out these beta cells, and then we take that blue gene that we identified as being the causal gene, and we perturb it, and we ask what happens. And so what we see when we do that is that these beta cells all of a sudden don't produce insulin anymore. So what we have now is we have a cellular model in which we perturbed a putative gene, and we now have a phenotype that correlates with type 2 diabetes, which is what we wanted. So now let's get super fancy and make a transgenic mouse, put in that SNP that we found, and see what happens to the mice. And we do that, and sure enough, we get mice here that have type 2 diabetes. So we have now, using sort of genomic information and a little bit of experimentation, connected ourselves all the way from the genome through cell types, variants, genes, functions, to like a meaningful understanding of disease biology, which we can then use to think about therapeutic strategies. Okay, well, that was super easy. So why haven't we figured this all out yet? Well, it's because that's not how this really works. This is how it would work if we had all the information and all the models in the world. But one of the big challenges is we often don't have the data that we need. So that magic book with all the cell types and all the structure doesn't exist yet. And so often, even if you have an atlas, say, to find your cell type, you might only have it at the tissue level. So maybe we have an atlas, but it's only for the pancreas as a whole. Well, all tissues in the human body are incredibly heterogeneous. And so there are many, many different cell types. And so if we had an atlas for the pancreas, well, that wouldn't necessarily help us very much for beta cells, which are a very, very tiny population of cells in the pancreas. And so it gets even more complicated, unfortunately. And even if we had an atlas for beta cells, the beta cells of young people are not going to have exactly the same patterns as beta cells of old people or beta cells of marathon runners. And so what this tells us is we need a lot more data. So this is, if you leave here with nothing else from my talk, is that this is why it's so important to invest in basic discovery science. So this doesn't have an immediate application. This is building atlases. But these atlases are essential for finding pathways to design drugs. So what we need more epigenetic data. We need to understand the function of the non-coding genome better. We need more expression data. We need to know what genes are on and where. We need this for more tissues and more cell types, especially the rare ones. And we need this, especially all of this information from a more diverse set of people. So we need it across the age spectrum from multiple races, and we need it from people that are exposed to different environmental exposures, because all of these things alter cell state. We have a couple of big projects that are happening. I'll just give a shout out to the most ambitious of these, which is the Human Cell Atlas, which the Broad is an active participant in, whose stated goal is to make a complete census of every cell in the human body, which sounds totally crazy, but I have no doubt that they'll be able to do it. So we're not there yet, but we're on the way. We need more models. So we don't have a magical refrigerator with all the cell lines we need. But we can go out into the population, for instance, and take samples from people at the high end of the risk spectrum and people at the low end of the risk spectrum, and we can ask what makes them different. Or if we don't want to wait for that genetic variation to just show up at our door, we can use genome editing tools, which are getting better and better all the time, and we could build that variation from the ground up. We need better assays. So many of the cellular phenotypes or behaviors that we want to measure, because they're related to the disease we care about, are very difficult to measure in the lab or can't be measured in the lab at all. And so what we'd like to do is to say, okay, so if we have this really complex cellular behavior, are there other phenotypes that tend to correlate with that? So can we instead use the shape of the cell or the gene expression signature? Or can we look at the proteins or the metabolites? And can we make a connection between this sort of more general phenotype, which is easy and fast to measure, and a more complex organismal phenotype? We want to do this at much bigger scale. So we want to use techniques like single-cell RNA sequencing or pooled optical profiling to do thousands of variants and thousands of cells in a single experiment. And then lastly, the biggest challenge is just that there are so many of these. So we have to really think intelligently about how we want to tackle this problem, because we can't just do these one by one. At the current rate of progress, it would take us well over 100 years just to get through the SNP trait associations we've already found, much less finding any new ones. So in addition, as a community, in addition to thinking about how we build better tools and better approaches, as we slowly tackle all these individual SNP trait associations, we're going to learn some rules of the game. And so we need to work as a community to take these rules as we learn them, build better computer algorithms, and make it so that we can do more and more of these experiments actually on the computer as opposed to in the laboratory, which is expensive and slow. And even though it'll put people like me out of business, it will make us much better to tackle the huge number of variants that we have. And that's all the time I have. I would like to particularly thank all of you for coming out tonight. And I'd like to give a special shout out to the amazing variant to function community here at Broad that I feel very, very happy to be able to be a part of. Thank you.
And thank you very much to all three of you for all your insights and for the discussion this evening. I'm going to invite JT, Alicia, and Amit to come and join us back up in the big red seats out here. And it's time for your questions. I will be running around with this microphone, so if you want me to come up and hand it over to you, just wave and give me a sec as I run out of breath and running upstairs and whatnot. Or we have a second microphone over here as well that you can come down to.
Thanks for some most informative talks. What's happening with Mass General's Biobank? Are you anywhere near the point where you can give feedback to patients on their risks? And would you give feedback?
Sure, so thanks for a great question. And for those of you who don't know, Mass General and Brigham actually went in together on what they call the Partners Biobank. It turns out actually about 90,000 people from the two hospitals have signed up and said they gave blood samples, they gave their DNA, and they said you can link this to actually our health information. And the question was, to what extent can we actually use that to give people some useful information back? Well, I'm excited to say that's actually a major focus for the next 2 years. We recently got NIH funding to actually start the sequencing of these individuals to find people with those rare monogenic mutations. And already the group has returned people who have BRCA1 variants related to breast cancer, high cholesterol variants related to heart attack, and others. I think these projects take a long time. I would say that the infrastructure is now built. We have the pathway to return results. And I'm excited that at Mass General, we're actually building out a new genomics clinical program that can actually house give a place for patients to go once they have this genetic risk information. So, so I guess I say thank you for your patience, but please stay tuned over the next 2 to 3 years.
That's great.
And there's some free advertising for the Biobank. Anyone? Yes. Excuse me. Sorry.
This question is for Amit. It's a very simple question. Can you talk more about how you obtained the polygenic score?
Sure. So the polygenic score for heart attack, as I mentioned, was really taking information from 6 million different places. And for each variant, we knew it increases people's risk by 2% or decreases it by 1%. And you might imagine that 2 variants are actually right next to each other. And each of them have a score of, let's say, 2, for example. So we basically used an algorithm developed at the Harvard School of Public Health to actually take that into account. So if there are 2 variants that both had a score of 2, they were right next to each other and highly correlated, we said, okay, let's give them both a score of 1. And we actually did that systematically across the whole genome. And so, and we published our score, it's available online, where it's literally just a list of 6 million variants, and each of those variants has a weight. And in terms of the calculation, it's actually simple linear algebra. So at any given variant, you could either have, like, for example, 2 A's, A and a G, or a G and a G, meaning different variants at that specific site. And we literally just say, for every time you have a G, add 1 point to your score. And we iterate that through that across the whole genome.
Thank you all for a very interesting talk and rather understandable. JT mentioned, not surprisingly, that variants occur in non-coding sites, and we need to better understand the functions of these non-coding regions. Now, I was under the perhaps naive impression that regions were called coding because they had specific functions, and non-coding regions were regions that had garbage or nothing of interest. But this seems to totally turn that around. And could you explain now how coding regions are defined, and are they still really properly called coding regions?
Yeah, that's a great question. So there's been this sort of term floating around for a long time of junk DNA, that most of your DNA is junk and the space between the genes doesn't do anything. And what we've learned is that because of the activity patterns I described and the way that the chromatin is remodeled in different cell types, is that what appears to be junk DNA in one specific lineage may in fact be crucial for differentiation or maintenance of another lineage. And so we don't really, I would be very hesitant to call any DNA junk DNA at this point. Coding DNA is still DNA that produces a RNA, which then goes on to produce a protein. There are also microRNAs and all sorts of other oddities. But yeah, I think the big takeaway is that that just because you don't know what a region of DNA does, don't assume that it doesn't do something absolutely crucial.
Up there.
Hi, I think this question's for Alicia. So your work reminds me a little bit of the work of David Reich at medical school. So I'm wondering, how do you, this is kind of a big question, but how do we meaningfully group people to perform the kind of studies you're talking about? Because if you look across time, what does it mean to be European? What does it mean to be East Asian? I'm curious to hear your thoughts about that. Thanks.
Yeah, that's a really, really great question. I think of human history and human evolution as sort of providing a lens into complex diseases, but what does it actually mean to be of European descent? I think is like a great question. What does it mean to be of European descent? On average means that you share more of your DNA with people who've been living in Europe for a long time, but that doesn't have a concrete meaning, and it also doesn't necessarily, a lot of the ways that we think about race and ethnicity are correlated with some of your environmental variables. That's not necessarily completely true for a given category. And if we think about broad strokes of race and ethnicity in the United States, there are large swaths of humanity that might have different interactions with different environments. So here in the US, you might think of African Americans being a fairly large part of our population, Hispanic and Latino populations being a pretty large part of our population. But that in and of itself is sort of, not a discrete bin. We don't necessarily have discrete Hispanic and Latino populations. There's admixture from, Europe, from the Americas, from Africa, in all of these groups. So it's very hard to necessarily say you are any one thing with your genetic data. What we typically do is just compare your genetic data with a reference panel, say from the 1,000 Genomes Project, where we have populations that look more similar to each other than with others, and ask what your genome looks like. So it's not a discrete and it's not a hard boundary. We sort of decide what your genome looks like with respect to the rest of the individuals in a given study.
How much work is being done on copy number variances, especially in regard to heart disease?
Yes, so the question is related to copy number variants, which are genetic variants which aren't just maybe a single misspelling of the code, but larger changes where, for example, an entire region of a group of base pairs is duplicated or deleted or so forth. So it turns out that actually those variants have actually been very difficult to detect using most sequencing technologies. In the work I mentioned that of, let's say, the 30 people who had monogenic mutations related to very high cholesterol, in 29 cases, it was related to a more traditional, like single base pair change. But we actually did analyze copy number variants as well. And we realized that 1 out of those 30 actually was missing the whole part of the chromosome encoding that gene. And so I would just say the algorithms just over the last 2 years have progressed to the point where we can reliably identify them. And I think once we reliably identify them, we can then associate which ones are associated with disease. So I would say, in broad strokes, the copy number variation field for diseases like heart attack is probably where the traditional genetic variant field was, say, 5 to 8 years ago. And I think that's the case now, but I would say it's rapidly catching up.
Actually, I'll exercise my moderator's prerogative and throw a question in. Is there, going off the theme of copy number variation, where you've got large portions of genome that are duplicated or lost, is there anything that your field, Amit, can learn from JT's where things like deletions and duplications and copies and whatnot can have a huge effect on whether or not a cell turns cancerous?
It's a great question. We're going to have to discuss this over beer at the reception. But
That's right.
I guess in broad strokes, I think we're very interested in folks like us working together, specifically to say, how can we profile people with a given genetic variant? Just like you profile a cell with a given genetic variant you create, what if you could go into the population and find someone who happened to have that exact same genetic variant, profile them, and use that to really understand the disease process? And I'll share just one anecdote, which is going back to, let's say, 2003, when we actually thought we knew everything about cholesterol metabolism. But it turns out that actually in about 2% of African Americans, and this highlights the importance of diversity, as Alicia mentioned, but 2% of these folks actually have not a risk mutation, but actually a broken copy of a gene that actually confers protection. So these folks, from the time they're born, actually have very low levels of cholesterol, and as a consequence for that, almost never develop heart attacks. And so this was in a gene that we didn't know anything about, was how it related to cholesterol or heart disease. But it turns out that then they would then be able to take that observation, go back into the lab, understand the mechanism, and then now I actually see a couple of patients in the audience. But basically now we have medicine that mimics that protective mutation, and they, even if they weren't born with it, we give that to them, and they can derive the same benefit. They're protected from heart attacks. So I think it's that kind of back and forth between identifying a genetic variant in humans that's associated with something interesting, or the vice versa, and going back and forth with the cellular models teams.
All right. Thank you very much, and thank you to all of you. It's 7:30. It's time for us to close up and move out to the reception. Thank you again for joining us tonight. Tonight's talk will be available on our YouTube channel. You can look forward by searching on Google for YouTube and Broad Institute. We encourage you to subscribe and get your friends to subscribe as well. And We hope you'll join us for the next Science for All Seasons talk, which will be scheduled for sometime in April or May. We'll send out all kinds of notices when that's ready to let you know. Thanks again, and have a good night.